Yeah, I will start with giving you some impressions of some volunteers participating in our exercise intervention studies. These were completely sedentary, elderly individuals between 40 and 60, and they were subjected to an exercise training for 6 months and then performed a half marathon. And I just tried to convince you that aerobic exercise can actually induce positive emotions. In fact, the psychophysiology of physical exercise encompasses different behavioral effects, and mood changes have been very often observed. Frequently, athletes report that they feel better, that they are calm, that they sleep better. In some rarer instances, they may experience very strong emotional effects referred to as the runner's high, but also in extremely strenuous exercise that can also be associated with dysphoria. Other effects encountered in exercise are anxiolytic effects, anti-nociceptive effects with the demonstration of elevated pain tolerance and also enhanced stress resistance. And the key modulator of these psychophysiological effects appears to be the amount of exercise, so the duration and the intensity of the exercise bout. The most propagated theory, as you may know, to explain these psychophysiological effects, especially the effects on mood and on pain tolerance, is the famous endorphin hypothesis, which postulates a causal role of central acting endogenous opioids released in the brain. In humans, it has been extremely difficult for technical reasons to really approach this hypothesis, and most of the research has been performed in a different compartment by looking at plasma beta endorphin levels, and very reproducibly it has been shown that the plasma beta endorphin levels increase during and after exercise bouts. There's a large inter-individual variability, and there is no good association between these peripheral beta endorphin levels and behavioral changes. In the animal literature, well, I should also say in humans, there's also some interesting work when you antagonize the system with naloxone eye drops. Some hints were given towards the central role of these endorphins in the context of exercise. In animals, there is clearer evidence regarding elevated CSF opioid levels, which have been shown in rats and in dogs. A number of studies showing altered opioid receptor occupancies, and very interestingly, by antagonizing the opioidergic system with naloxone, withdrawal symptoms were elicited in animals which were previously subjected to a very intense exercise training, withdrawal symptoms that resemble morphine pre-treatment. Now, we were the first to probe the endorphin hypothesis in the context of exercise in humans by using a PET ligand activation approach, using the PET tracer F18 FDPN, which is a dipronorphin derivative, which has a similar selectivity to the mu delta and kappa opioid receptors. And here you see an example of the pattern of ligand binding you find typically in healthy controls. The idea of this ligand activation approach is that the binding of the PET tracer, shown here in red, at the opioid receptor sites will heavily depend on the amount of endogenous opioids. And our specific hypothesis was that after running, we would observe a reduction of the tracer binding due to enhanced endogenous opioid release as compared to a control condition. And we also had a specific hypothesis regarding the areas where we would identify possible changes in opioid receptor binding, informed by this very nice work by Kringelbach and Barrage, showing that the conscious aspect of pleasure mediated by opioids and endocannabinoids is related to changes in frontal and limbic structures, including the orbitofrontal cortex, the ACC, and the insular cortex. So in this study, due to restrictions from the radiation protection authorities, we were only allowed to study 10 male athletes around 37 years with a training duration of something around 9 hours per week. Body mass index was 23, and they were subjected to 2 consecutive PET scans 4 weeks apart. And we quantified the distribution volume using basis pursuit denoising, which is a spectral analysis approach implemented by Roger Gunn in the London group. And we performed 2 PET scans, 1 PET scan at rest, no exercise on the day of the examination and on the day previous to this PET scan, and another PET scan immediately after a 2-hour outdoor jog. And we also performed visual analog mood scales before and after the interventions, and we did drug screenings for opioids in the urine. The duration of the run was 115 minutes at a pace of 11 kilometers per hour. The distance was 21.5 kilometers, which is about 13 miles. Heart rate 144 at a rest heart rate in athletes, typically very low, of 52. And here you see the individual increases in the euphoria ratings before and after the run in the 10 subjects, and you can see that there's a marked increase in the perceived euphoria state. If you look at the group results, there was a significant increase post-run as compared to the pre-run condition and also to the rest scan 4 weeks apart. And we also did some additional longitudinal follow-ups on these euphoria scales after a 2-hour run in these 10 subjects, and you can see that this is a prolonged effect which levels off slowly and is maintained over several hours. Upon imaging, we were able to identify significant changes and only reductions in the FDPN binding, and they were localized mainly in orbitofrontal areas, in the anterior insular cortex, and in the ACC, in particular in the rostral part of the anterior cingulate cortex. And this fits very nicely to the hypothesis we had based on this work by Kringelbach and Barrage, where these effects were suggested based on mainly animal work. And this effect here in the rostral ACC was inverse, just the opposite effect, compared to another study using a mu-selective opioid tracer, C11 carfentanil by K. Zubieta's group, where they induced experimentally a sustained sadness state. And in this study, these authors observed an increase in the opioid binding in exactly the same area. So these are opposite effects depending on the emotional state induced by the intervention. In our study, we observed an inverse correlation between the binding of the FDPN and the VAS euphoria ratings, meaning that the higher the euphoria ratings, the lower the binding of the tracer in that specific area. And this was not only the case for the rostral ACC, we also observed these effects in the orbitofrontal cortex, the insula, and also some frontal areas. So in conclusion, this was a first proof of principle study, ligand activation in exercise of the opioidergic system. We observed a significant reduction of the FDPN binding in widespread brain areas, but mainly in limbic and prefrontal areas. Notably, no increases in FDPN bindings, and these findings were interpreted as being indicative of endogenous opioid release related to the perceived elevated mood state after a strenuous exercise. It took about nearly 10 years until 2 further studies came out from different groups in Turku and in Tokyo. And these were further advancements in this field because they used a mu-selective opioid tracer, and they also compared different exercise types or intensities. This first work here, published in Neuropsychopharmacology 2017 by Sanyoki and co-workers, as I said, used C11 carfentanil. It's a selective high-affinity agonist radioligand for the mu-opioid receptor. These are known to be directly linked to positive reward and euphoria and mediate effects both of endogenous and exogenous opioids. So they are really at the core of what we are investigating here. And this work group studied 22 healthy young men around 26 years, and they performed 3 PET scans, one at rest without any physical exercise prior to the PET scan. Then, after 60 minutes of moderate intensity, continuous cycle ergometer training, and in a subgroup of 12, additional high-intensity interval training on a cycle ergometer. First, the MICT session. This was really an aerobic exercise. The mean workload was around 160 watts, and the average heart rate 143, and this was associated with positive effect and euphoria ratings, which were higher than in the very strenuous condition, the HIIT. This HIIT consisted of a number of sprints for 30 seconds on the cycle ergometer as an all-out cycling effort until exhaustion, with 4 minutes of recovery. The exertion level was higher than in the MICT. It was also associated with higher negative effect, and the binding of the carfentanil showed a significant widespread reduction in different brain areas in the range of about 19%. Here I show you where these effects were localized compared to the rest condition. Again, very much resembling our previous work in the orbitofrontal cortex, in the insular cortex, in the ACC, and also in some frontal and parietal areas. Here you see the 2 largest clusters showing this dose relation, which I find extremely interesting. So the black bar is the very strenuous exercise, the HIIT, and you see this for both clusters. So this indicates that there is a dose-dependent effect. And they also observed an inverse relationship between perceived euphoria or changes in euphoria and changes in the binding potential of C11 carfentanil, meaning that the highest gain in euphoria was associated with the largest decrease of the binding in the area shown on the slide. So this study showed very nicely that high-intensity interval training elicits mu-opioid-specific binding reductions that are, again, indicative of endogenous opioid release in areas which very much resemble our initial study. They also observed no increases in the binding and no significant change in the aerobic exercise condition. So decreased mu-opioid receptor availability following HIIT but not MICT indicates a dose effect of endogenous opioid action at mu-opioid receptor sites in response to physical exercise. And there's a second study which also came out in 2017 by Hirura et al. In the International Journal of Sports Medicine. Again, 2 exercise conditions and again C11 carfentanil. A small group of recreationally active young males. 2 conditions, a heavy condition defined as 60% of VO2 peak individually determined, and a severe condition with 80% of VO2 peak. And as you can see on the right, These two scans were performed in randomized order, or two conditions were performed in randomized order, and they were, and before and after the PET scans were performed, so a total of 4 PET scans per subject. And they not only observed significant changes in the profile of mood scale with significant reductions in mood ratings for depression and anger, they also found a reduction of the tracer binding, again in the rostral ACC, in the anterior and posterior insular cortex, and also in the cingulate areas. They also observed tendencies which were not really significant as main effects of intensity and an interesting interaction effect in the pituitary, but I will not further comment on this because these were only strong trends and did not reach the level of significance. So in conclusion, I think there is converging evidence from 3 independent PET ligand activation studies to support the endorphin hypothesis in humans. The effects appear to be driven mainly by mu-opioid receptor-specific effects. The regions involved are mainly prefrontal and limbic areas, and there is a relationship to perceived mood changes, and as shown by these 2 newer papers, which I find really interesting, there appears to be a dependence on exercise intensity. So I would like to thank you for your attention. I would also like to thank our sponsors, all my collaborators in the first study, and also our volunteers who participate in these studies. Thank you very much.